Hemolytic anemias all share one core problem. Red blood cells are being destroyed faster than they should be, shortening their normal lifespan. When this happens, the hemoglobin that spills out gets broken down into various byproducts, each of which leaves a detectable footprint in the body. The first and most clinically recognizable sign is a rise in unconjugated indirect bilirubin in the blood. Because this form of bilirubin has not yet been processed by the liver into a water-soluble form, it cannot be excreted into the urine. This results in a type of jaundice called acholuric jaundice. The skin and eyes turn yellow, but the urine stays clear and bilirubin-free. Over time, the sustained elevation of bilirubin can cause it to crystallize in the gallbladder, forming pigment-containing gallstones as a long-term complication. Second, urine urobilinogen levels rise, since more bilirubin is being delivered to the gut and eventually recycled. Third, when red cell destruction happens directly inside blood vessels, a process called intravascular hemolysis, free hemoglobin spills into the bloodstream and then into the urine, producing hemoglobinemia and hemoglobinuria. At the same time, a related compound called methemalbuminemia forms, and iron deposits in renal tubular cells cause hemosideриnuria. Fourth, serum haptoglobins disappear. Haptoglobins are proteins that normally circulate in the blood, with the sole job of grabbing free hemoglobin and removing it safely. During hemolysis, these proteins get consumed so rapidly that they become undetectable. Importantly, in intravascular hemolysis, such as during a transfusion reaction, serum hemoglobin does not rise until all available haptoglobins have been fully exhausted. Fifth, systemic iron deposition, called hemosiderosis, develops as iron from destroyed cells accumulates throughout the body. In summary, excessively rapid destruction of red blood cells (RBCs), a process known as hemolysis, leads to hemolytic anemia, characterized by decreased red cell mass and tissue oxygen delivery, elevated unconjugated indirect bilirubin from increased heme breakdown, and, depending on the location of destruction, findings such as jaundice, splenomegaly, reticulocytosis, hemoglobinemia, and hemoglobinuria. Hemolytic anemia results when the rate of RBC destruction exceeds the marrow's ability to compensate, leading to low hemoglobin and classic anemia symptoms: fatigue, pallor, dyspnea. Findings usually include increased reticulocyte count, elevated lactate dehydrogenase (LDH), decreased haptoglobin, and increased unconjugated bilirubin. Hemoglobinemia and hemoglobinuria indicate intravascular hemolysis. Jaundice and splenomegaly may develop due to increased bilirubin turnover and hypertrophy of the reticuloendothelial system, spleen, liver, from increased red cell clearance.